Apologies for not speaking in Italian, but for me it's very difficult to speak in Italian, in general, but especially about science. But I think English is the language of science anyway, so maybe it's good for you to hear presentations in English as well. So I will be talking about gene network inference. First, I would like to give a small overview of my lab, of my group at CRS4, and my group is called Systems Genetics, and well, we also call this the ALF lab. ALF is my nickname, and there are three young people working with me, which are Andrea Pina, Nicola Soranzo, Vincenzo Di Leo. So what you see below, is there a pointer? Is there a laser pointer? Laser pointer. Well, what you see here below is essentially the goal of my research summarized in just one figure. So we are very interested in how the genotype and the environment are turned into a phenotype. A phenotype like the way you and I look. And okay, there's an improvement since yesterday. But so we would like to find out what are the regulatory systems that mediate, the conversion from genotype and phenotype into the phenotype of organisms. So the main activities of my lab consist of three parts, research. The biggest part of my research is gene network inference, and this is where I will talk mostly about today. Then we developed the Systems Genetic Simulator. It's a program that can simulate data that can also be produced in a laboratory, and we do this in order to be able to evaluate algorithms that we develop to analyze these data. So if you simulate data, you know exactly the model that you start with, so you can compare what you have, what comes out of your analysis with the ground truth. And at the end of the talk, I will put my work a little bit in the context of medical research, and I call this differential networking indices. So this is essentially trying to figure out what went wrong in terms of regulatory networks between healthy tissues and, for example, tumors. So I want to find out what are the differences in the regulatory networks. So the overview of the presentation is, first I give you an introduction to gene networks. So I don't know what your backgrounds are. Are you biologists or mathematicians, engineers, or maybe all of them? Anyway, so I will give a very basic introduction to biology. So sorry if you are a biologist, you will hear some things that you probably already know. Then when it is clear what gene networks are, I will talk about the gene network inference. So identifying gene networks from experimental data. And I will end with differential networking indices. Okay, so first, what are gene networks? So this is the basic introductory slide about the central dogma of molecular biology. So it shows that the DNA containing the genes is just sitting there in the cell, containing the information, encoding the proteins, which are the workhorses in the cells. The proteins are the molecules that actually do things. They catalyze reactions. They regulate the activity of genes. They form the building blocks. But so between the DNA and the proteins, there's a molecule called RNA. This is kind of a messenger. And so this molecule is somewhat central in my presentation because this is a measure of gene activity. So if a gene is active, it will be producing a lot of RNA through this process called transcription. So the more RNA there is, also the more proteins there are translated from it. So when I refer to gene activity, or when I refer to gene networks, I am actually talking about relationships between RNA molecules. So there are many different ways to experimentally measure these molecules. Especially, there are a lot of data sets available from the DNA sequences. So there are many organisms the DNA sequence has been determined. For example, also for human, and now they are also sequencing individual humans, like thousands of individual humans. So we have a lot of the sequence data, but sequence data alone is just the information that we don't know what. So the information that codes for proteins, but we don't know how these proteins work together in order to give rise to a phenotype. Also, the messenger, the RNA, can be measured with high throughput technologies. For example, microarrays. So in which each spot, the intensity of each spot in this array gives you an indication of the concentration of the RNA. And currently there are also these new machines where next generation sequencing machines, and you can also use this to determine messenger RNA concentrations just by sequencing all the nucleic acids in the cell. So actually what you would like to measure are the proteins themselves, because these are really the things that do things in the cell. But although there are experimental technologies to measure them, they are not as high throughput as the other technologies. So in a way, I focus on the RNA because there is lots and lots of data available on RNA, and I use them kind of as an approximation to the concentration of proteins. So this is a typical DNA sequence. So now from here we need to figure out how our phenotype is established. So what are gene networks? So each gene, so a part of the gene encodes for a protein, but there are also sequences in front of the gene that play a role for a regulatory role. For example, here you see a gene, and here's the promoter region, so the regulatory region, and there are many proteins binding to this region, and as a result, they activate the gene, so they start the transcription process. So what you see here, there are a couple of proteins which are activators, and there's a protein which is a repressor. And so I prefer to convert this picture into something like this, in which just the target gene is a node in a network, and also the inputs are represented as nodes. So this is a much more simplified picture than so this looks a lot more simple than all this molecular machinery. But you can represent all the regulatory relationships between the genes in this way. And then you get something more complicated again. But at least this is a description of all the regulatory relationships inside a living cell, and not just of one gene. It's even a little bit more complicated because genes, so the RNAs, they don't interact directly with each other, but so they produce, for example, a protein, which is a factor that activates or inhibits another gene. Or in some more difficult, more complicated cases, a particular gene encodes a metabolic enzyme that in turn catalyzes a metabolic reaction, and the product of the metabolic reaction regulates the activity of another gene. Now, if we only look at gene expression data, so the RNA levels, we don't observe the metabolic space, we don't observe the protein space. So what we actually can infer is a network, which is actually a projection of all this biochemistry on top of the gene space. So for example, gene 2 regulates gene 4, although it goes all the way through this whole cascade of reactions. So gene networks are rather abstract representations of the biochemistry underlying the interactions. So essentially, so if you have a simple representation as a network of these complicated regulatory processes, you can also represent them in a mathematical form, which is a matrix. And for example, the top matrix is called an adjacency matrix, and essentially there are ones in positions where there is an interaction. For example, in the first row of the top matrix, I wish this was a little bit longer, but you see that the fifth column has a 1, and that corresponds to this effect of this gene 5 on gene 1. So in this way, the zeros mean no interaction, and the ones mean there is an interaction. So this is a mathematical representation of a gene network. On the other hand, you can also represent it as a weighted network. So you can actually, instead of using ones or zeros, you can use real numbers to indicate the strength of interactions. But yeah, so this mathematical representation allows us to perform all kinds of mathematical analysis on the gene networks. Okay, so the data that is required to infer gene networks is gene expression data. So I already said that this is the microarray, but nowadays you can also use this next generation sequencing. But in order to infer gene networks, you really need a lot of data. I mean, gene networks can consist of thousands of genes. So if you want to find a network between thousands of genes, you need a lot of data. Also the data can be represented as a matrix, as a P by N matrix, where P is the gene, so all the rows are the number of genes, and N is the number of observations, or the number of patients that these genes have been measured in, the number of patients or number of individuals. Okay. So now we have some idea what gene networks are. So let's now go to how to infer gene networks. So this is essentially the basic problem of inferring gene networks. So it's mathematicians call this an inverse problem, or engineers tend to call this a system identification, because the idea is that you start by making observations of a system, in this case a gene network, and then you try to figure out what is the underlying system that produced these observations. So, having made measurements of gene expression, what is the most plausible network that can produce the data, observed data? And so typical methods that we use is correlation, partial correlation, regression, ordinary differential equations, graphical Gaussian models, and perturbation analysis. And there are many, many more methods proposed for this problem. So essentially, going back to this slide, inferring gene networks is to find out where in these matrices are the non-zeros. So which genes interact with which other genes, so, or identifying the zeros. So finding out where there is no interaction. So that's the main problem. Okay, so there are different ways. There are different types of experimental data that can be used. For example, there's a data type called observational data. So observational data are repeated measurements of a given tissue, cell type, without an experimental intervention. So without any change. So without inducing any experimental change. So you could imagine a study on liver gene expression, and you take liver samples from a group of people, and you measure their liver gene expression. So you don't make any experimental interventions. You just look how gene expression at the levels of gene expression in a particular tissue. So you could apply network algorithms on this, but it allows only for inferring undirected networks. So since you don't make any experimental interventions, it's very well. It's impossible, almost impossible to actually find out which genes regulate which other genes. Only that they are, for example, associated with each other. With perturbation data, you create targeted perturbations, and then you measure the dynamic responses in the cells. And this allows for inferring directed networks. So observational data looks like a bit like this. So each point in this plot is a particular individual, and there are so the levels of two genes are plotted against each other. So this is a patient in which the gene A expression has this value and gene B expression has this value, and you see in the first one you see that there's if gene A is low, also gene B is low. If gene A is high, also gene B is high. So there's a strong positive correlation. So in this case, you could say, okay, given the fact that there's a correlation between the two, we can draw an edge between them. And in another case, for example, gene A versus gene C, you see that there's no correlation at all. And in that case, you wouldn't draw any edge between them. So in this way, you can create what are called co-expression networks. But of course, there's a drawback on this because you get a correlation network, but correlation, of course, it doesn't equal causation. For example, I put here this. I don't know. Can you read this? Because it's very small, but. This is an example of the where it can go wrong with transferring or interpreting correlation as causation. It's a plot of the number of pirates in the world, versus the global average temperature. And, well, you see that this is a very strong correlation, but that obviously doesn't imply that the more pirates there are, the higher the global temperature. Or maybe it does, but... But for example, if we have this undirected edge between two genes, this could imply that gene B regulates gene A. It could imply that gene A regulates gene B, or there's a third gene that regulates them both. So this creates correlation. So the correlation networks have the drawback that we cannot find out which genes regulate which other genes. In other words, we cannot really infer gene networks from this type of data. To infer directed gene networks, so to infer the causal relationships between genes, you really need to have perturbation data. For example, there are steady-state perturbation data. So imagine you have a particular cell line growing into in a flask, and you measure the gene expression in this wild-type state. Then you create a strain of this organism in which you overexpress a certain gene, and you measure again the gene expression. You overexpress another gene, you measure the gene expression, and etc. until you have overexpressed and measured the responses to all the genes in the genome. Or you can use time series data, in which you apply some stress to, let's say, a cell culture, and see how the gene expression changes in time. So from time series, you're also able to find out the causal relations between the gene activities. So most of my work actually considers perturbation data. And so the method that I'm going to talk about now it's called a perturbation analysis, in which you need the following type of data. So you need steady-state gene expression data for a wild type, just what I was telling you about, and then the systematic single gene knockdowns or overexpression. So it requires small perturbations, and so knockdowns can be established, for example, by making heterozygous knockouts. So maybe you know that most organisms, they have two chromosomes. So each chromosome, so diploid organisms have two chromosomes for each chromosome. If you would knock out the gene copy on only one chromosome, you still have one intact. But you have reduced the number of gene, so you have reduced the gene doses by half. Overexpression, for example, could be by adding plasmids with an extra gene copy. So then you increase the gene dosage, so you would have higher activity of the gene that is on the plasmid. So this perturbation analysis is actually very simple, because it works like this. So assume that this little network is this is the real system. So what happens if you would perturb the first gene? What other genes will you see respond? Well, you see that the first gene regulates the second gene, which in turn regulates the third gene, which in turn regulates the fourth gene. So you can imagine that these three genes will respond to the perturbation in gene 1. So obviously we will find the second gene, but also additional genes which are downstream of the second gene. If you perturb the second gene, you get the same. You get the third gene and the fourth gene. And if you perturb all the genes, you get the network like this. So this network contains the direct interactions between genes, but also the indirect interactions. So transitive interactions. So here comes the trick. Is that so? This is the data matrix that represent the data matrix represent the changes in gene expression with respect to the wild type. So, for example, here in the first column, these are all the genes that respond to the perturbation in the first gene. So you see that gene 5 doesn't respond because it's not downstream of gene 1. So in the second experiment, only 3 genes respond, etc. So if you perturb X4, if you perturb gene 4, only itself will change because there is no outgoing relationship. And there's a very simple mathematical relationship between the actual network, the weighted network matrix, and the data matrix. This is by taking the inverse of the data matrix. I don't know, are you all familiar with what an inverse of a matrix is? So everybody's familiar with what the inverse of a matrix is? Okay. Anyway, so this is all based. So this simple relationships comes from a linear modeling approach. So I will not go through the details of this, but the final result of this approach is this relationship. But the idea is that you start by the assumption that gene networks can be described by nonlinear differential equations, which we then linearize. And using steady states, you can convert it into an algebraic relationship. If you are interested in this, then maybe you can ask me a question about it later. Or you can also read a lot more in these papers. It's all described very well in these papers. Okay. So because of this linear framework, we actually require very small perturbations. The small perturbations are experimentally very difficult to create. And most of the experimental data is actually not heterozygote knockouts, but full knockouts of genes. So there are libraries created for example, yeast, in which they have created a knockout, so they have removed both copies of each gene for all the genes. So the previous method was, in a theoretical sense, quite interesting, but it's not very applicable to real data sets. So we decided to develop a method that is actually more readily applicable. And again, the type of data is very similar. We need the wild type, so the wild type gene expression, and instead of systematic single gene knockdowns or overexpressions, we can deal with complete knockouts. And in this case, we start out by quantifying the effect of a knockout of one gene on the gene expression of another gene by using a z-score. So we estimate a weight for an edge from gene I to gene J. So this is essentially a change in we use the change in the RNA level and the gene expression level, XYJ, of gene J after knockout of gene I. So this XIJ is the value, the gene expression value of gene J after knockout of gene I. And so we subtract the mean of gene J over all, so the mean taken over all the perturbations, and divide by the standard deviation over all the perturbations. So essentially, so if this z-score is high, that means that the perturbation in I is very important in the change in gene J. So this is the first thing that we do. But again, we have we quantify the importance of perturbations on the expression levels of other genes. But again, we have the same problem as before. There are direct edges, but also there's transitive causality. So mediated genes will also respond. So for example, consider this little gene network example. You see that gene 1 affects gene 2, and gene 2 affects gene 3. But we also find an edge going from gene 1 to gene 3. But now you might wonder, does that edge really need to be there? Because if it is not there, you could still expect that gene 3 would respond to the perturbation in gene 1, because there is an indirect path from gene 1 to gene 3. So essentially, we take a full network determined by some z-score threshold. So we say z-scores that are above a certain threshold, they create edges in the network. And so we determine all these edges that are not really essential for the reachability between genes. So this one, that edge between gene 1 and gene 3 is not essential for the reachability from gene 1 to gene 3. So we sparsify the network, we remove edges in the network that are not essential. Again, if you think this is interesting, you can read further on this. So the slides will be available on the website. Yeah? Okay, so this just a nice figure. This last method that I described. Some other researchers have performed a study of many different algorithms and our algorithm as well, to see how it performs on simulated gene knockout data sets. And so here, this is the PINA approach. This is the one that I just described. It is really the best performing algorithm compared to many other algorithms proposed for gene network inference from this type of data. Okay, so the final part of the talk is about differential networking in disease. So, disease studies, it's all about finding differences between two groups, for example, groups of healthy tissues versus tumor tissue, group treated with medicine versus not treated by medicine, group with tumor stage X versus tumor stage Y. Well, of course you can already by eye see that these lungs are pretty different. But to really go into the molecular detail of what the difference is between the disease and healthy, you can also do this on the gene expression level. And typically, this is done by testing for differential expression. So this goes like they take one gene and see whether the mean has changed between healthy and sick. And so they perform a test to show that this mean is different from the mean in the mean in the control group, the healthy group, is different from the mean in the sick group. But what I really would like to do is the following. I call this differential networking. So instead of testing, comparing individual genes, I would like to gather data from sick tissues and gather data from healthy tissues, apply these algorithms that I just have described, and then actually identify networks, and then do the comparison based on the network, and to try to identify parts of the network that have been impaired, or part of the networks that have been enforced. So really to understand the regulatory systems that are underlying diseases. So I wrote this review in the journal Trends in Genetics. Essentially, it's about going from differential expression to differential networking. But unfortunately, it is not really. We are not really there because, of course, in humans you cannot do this perturbation. You can do perturbation experiments. You cannot just create a whole bunch of humans with one gene deleted. That is not ethical, and also it's very time-consuming. So most of this will be on the level of co-expression. So again, what you see here is this is gene A expression in healthy subjects. This is gene B expression in healthy subjects, and gene A and gene B in healthy subjects are highly correlated. On the other hand, this is gene A expression in sick subjects and gene B expression in sick subjects. And you see that the correlation has completely disappeared in the sick subjects. So in terms of association, there might be something interesting going on in the regulatory network involving these two genes. So although we aren't really yet at differential networking, but we can do a lot of differential co-expression analysis. So this is something that we are working on right now. In this case, we don't infer networks from data, but rather take networks that have been published in the literature in databases. And so the idea is that there are known regulators, and their targets are known. So if we would have a healthy regulatory system, so these targets will be co-regulated, and there will be correlation between the gene expression activities of these genes. On the other hand, if in a particular sick regulatory system, these effects are no longer present, then the correlation disappears, because the coordinating factor has disappeared. So the idea is that You take healthy gene expression, you calculate the correlation matrix. You take sick gene expression, you calculate the correlation matrix. And then for each of the correlations, you take the correlation between gene I and J from the healthy minus the correlation between the same two genes in the sick. And so this is the Euclidean distance, but it's for a given set of genes. It summarizes the correlation changes in that set of genes. So if we would find a very strong change in a particular set, that might tell us if a certain regulator is impaired in a particular disease. So, so the network that we use is so it's collected from existing databases. So we created a new database which we call GenXBRAC. It's a database that we think contains almost, well, let's say, all that we know about gene regulation. It's in the database. Of course, there's also a lot not known, but at least what we known so far is in this database. So there are, for example, so transcription factors of proteins, regulatory proteins, target relationships, lots of them. Also microRNAs. MicroRNAs are small RNAs that actually are not translated into proteins, but do have regulatory functions themselves. So this is a network that we created this big network to be able to search for dysregulated regulators in disease, and so we evaluated this on simulated data. So essentially, we simulated data with a nonlinear differential equation. So these simulations were based on only the regulation by the microRNAs, which act by increasing the degradation of the transcripts. So there's no, I cannot reach it. you will have less problem. But anyway, so it's so in this model, the RNAs are produced by a fixed rate and are degraded based on the concentrations of their affecting mRNAs. So, and what we did is, so this is the measure that I showed before, and we also tried another measure. So the one is called an L2 measure, the other one is called L1 measure. And so we evaluated how well this method could identify the microRNAs that we knocked out in our simulations. So we would simulate data from a healthy person, then we would randomly knock out 10 regulators and simulate data so without these 10 regulators. And the idea is that we should be able to identify which 10 have been knocked out. And actually, so without going on what precisely these measures are, you see that you can see that the performance is quite good, but it decreases so if the contribution of the variance in the gene expression due to the regulator decreases, it gets harder to identify these the regulators that have been knocked out. which is in a way quite obvious. So we also applied this to a real data set. So we tried to identify this, which microRNAs had been impaired in lung cancer. And these are the results. We identified a bunch of microRNAs, and actually some of them, so most of them, there's nothing known about their function. But from the ones that have a known function, they're all related to processes involved in lung cancer. For example, here lung cancer tissue. So it seems that what we identified really matches what we already know. Plus, we identified a number of potential microRNAs that might be related to lung cancer. So we generated a set of new hypotheses. Okay, so this was essentially my talk, and I would like to end telling you about an initiative that I am involved in. It's a collaboration between IBM and Philip Morris. And what I would like to do is to start what is called a collaborative competition, which means that they release challenges, they release data in which you have to make predictions and submit to them, and they know the true answers, so they can evaluate your algorithm. And so in this way, it is some kind of competition, but on the other end, of course, it's a collaboration between many scientists that work on the same topic and will work on the same data set. So in this, so there have been some challenges released, and okay, so this is not concerning gene network inference, but it's about disease classification from gene expression data. So they provide, for example, Philip Morris is a cigarette company, so they are mostly interested in lung diseases. But so for example, they provide data on healthy lungs, and they provide data from lungs with a particular disease, obstructive pulmonary disease. So you get a certain amount of data, a training set. So they tell you these are gene expression samples from healthy, these are gene expression samples from this sick lungs. So you need to learn a model that can predict, that can distinguish them based on gene expression data. Then you get a test set, which you have to classify, and then these predictions will be compared to the actual classification that is known by the challenge organizers. So I wanted to mention this because I don't know, maybe some of you are interested in participating. So the first prize is $40,000 in research money, and of course, if you perform well, if you win this challenge, I mean, it's good for your reputation. People will trust more in your algorithms. So if you're interested in this, there are some flyers that on the desk there. So go ahead and grab them. And that's all for me for today. So I'm open for questions. If you want to ask questions in Italian, it's good.